Hello and welcome to video 25 for Mr. McGeehee's 9th grade biology class at Arizona School for the Arts. Today we're going to examine the structure of DNA and chromosomes. Let's start by reviewing a few basics about DNA. Recall that DNA is a long molecule made of nucleotides, and that the nucleotides come in two types: purines, like adenine and guanine, and pyrimidines, like cytosine and thymine. Purines have two rings in the nitrogenous base, where pyrimidines have only one. The backbone of DNA is made by covalent bonds between sugar and phosphate groups of the nucleotides. Along this backbone, the nucleotides can be joined in any order. In the 1950s, a scientist named Erwin Chargaff observed a curious relationship between the four nucleotides. By studying many different organisms, he found that this pattern was similar for all living things. He discovered that the amounts of adenine and thymine in DNA were roughly the same, as were the amounts of cytosine and guanine. This pattern was found in every organism he studied, although he didn't have any idea why this occurred. In short, Chargaff's rules state that A equals T and C equals G. Also in the 1950s, Rosalind Franklin began to study the complex three-dimensional structure of large molecules like DNA. She used a technique called X-ray diffraction. By shining X-rays through samples of crystallized molecules, Franklin produced a complex pattern of spots on photographic plates. By analyzing these patterns mathematically, Franklin could then infer the shape of the molecule she was studying. This was a brilliant technique that required hours of calculations, without the help of computers or even calculators. Can you imagine Hector doing something like this? The images that Franklin produced were extremely detailed. The X-shaped pattern in the middle of this famous image of DNA indicated to her that the molecule was a coiled helix. Meanwhile, scientists Francis Crick and James Watson were studying the structure of DNA by building large 3D models in the hopes of figuring out how all the nucleotides in DNA were put together. It was a bit like making a huge 3D puzzle without the benefit of the image on the front of the box. They examined a copy of Franklin's X-ray diffraction images. When they did, they realized that DNA was in the shape of a double helix. And although Franklin's images described the structure of DNA, they did not help to understand what forces held the helix together. Watson and Crick discovered that hydrogen bonds between nitrogenous bases hold the two sides of the helix together. Further, Watson and Crick discovered that hydrogen bonds could form only between certain nitrogenous bases. They found that A would only bond with T, and G would only bond with C. This principle explains Chargaff's rules. DNA is stored as long molecules called chromosomes. Prokaryotes store their DNA as a single loop in the cytoplasm. They have one chromosome. Eukaryotes have as much as 1,000 times more DNA. It's stored as many chromosomes in the nucleus. Chromosome number varies between species. This image shows the DNA of E. coli bacteria unraveled outside the cell. The chromosome of E. coli is 4.6 million base pairs long. Although when stretched out the chromosome is only 1.6 millimeters, this is many times the length of the entire cell. In order to fit it all inside the bacteria, it has to be folded up to 1,000th its length. A human cell contains almost 1,000 times as much DNA as a bacterium cell. Inside every human cell is DNA that is more than 1 meter long. During metaphase, this DNA is condensed into rod-shaped chromosomes, seen here. Eukaryotic chromosomes contain both DNA and proteins. They're tightly packed and coiled. This substance is called chromatin. A single strand of DNA is wound around bead-like proteins called histones. Clusters of these histones are called nucleosomes. These are coiled to form long chromatin fibers, which are further coiled into what are called supercoils. These supercoils are wrapped and folded into the larger structure that we call the chromosome. During most of the life of a cell, the chromosomes are dispersed in the nucleus and are not visible. When the cell divides, however, the chromosomes condense and they can be seen using a light microscope. That's the end of this video. Please make sure you complete the content questions and be ready to review all of this material in class. And here's your bunny of the day.